He disappeared. Okay, I'd like to thank Vitor, Dmitry, and all of the organizers for putting together this workshop and for the invitation for me to speak here. I'd like to share with you some of our findings that address the issue of multiplicity of folding pathways. I'm talking about the pathways through which proteins and nucleic acids undergo their conformational transitions. Let me first explain what motivated us to think hard about this issue, the issue of multiplicity of folding pathways. I'll begin with an observation. The observation is that, not unlike geographic landscapes, the energy landscapes of biological macromolecules are complex. They are hyperdimensional, they are rough and hierarchical. In the light of this complexity of the energy landscapes of proteins and nucleic acids, it is natural to expect that folding and binding transitions should be commonly mediated by multiple pathways as opposed to a single route. Of course, at a given set of environmental conditions, only a small subset of these pathways, or maybe just one of these pathways, will dominate the conformational dynamics. However, when the external conditions change, the pathway dominance may change as well. If this all is true, it means that biological macromolecules have the freedom to switch to an alternative pathway in response to external perturbation, and to take that alternative pathway instead. It also means that this freedom to switch folding pathways in response to the change in the environment may have some important functional consequences for the biological function. Let's take this largely qualitative picture and turn it into something more quantitative by first defining some key terms. First, the energy landscape. So the energy of even the simplest biological macromolecule, such as a small protein, is a function of not just one or a few, but of tens of thousands of degrees of freedom, 3 degrees of freedom per each of the thousands of atoms that the protein is made of. If we were to visualize this energy function as a graph, we would have to imagine a rough hypersurface in a 1,000-dimensional space. So the term landscape, the energy landscape, seems indeed very appropriate. This is our attempt to visualize the energy landscape of a small protein by drawing just a fragment of this landscape in the space of just not 1,000, but just 2 coordinates. Next, pathways. A statistical mechanical definition of a reaction pathway is an ensemble of trajectories in configurational space that connect the initial and final conformations of the biomolecule in our context. The complexity of the energy landscapes of biomolecules and the resulting multiplicity of its folding pathways leads us to ask the question, which is the central question of this talk. The question is, how can we tell if the given conformational dynamics, folding and folding, binding and binding, zipping and zipping of proteins and nucleic acids, is mediated by multiple pathways? To attempt to answer this question, we can try to take advantage of single molecule force spectroscopy experiments that were already introduced in a number of talks at this workshop, and we will hear more from Michael Woodside and possibly others about this. The power of single molecule force experiment is that one can apply a controllable force to pull the molecule apart and to record the response of the molecule to force in real time. One of the outcomes of the pulling experiments is a set of conformational trajectories. The extension is a function of time. We generated these trajectories from Brownian dynamic simulations. These trajectories nicely visualize our molecule dwelling for some time in the folded state and then escaping over the barrier into the unfolded state. So now that we have access to individual conformational trajectories of our molecule, just like one would have in an experiment, can we tell if these trajectories come from one or multiple pathways? Well, unless one of the pathways is marked by a distinct trait, such as an intermediate state that can be reliably resolved in a broad range of forces, clearly not the case here, and usually is not the case in the experiment. There is really no way to tell whether these conformational dynamics are governed by one or multiple pathways. Now let's see if we can make the task a little bit easier, and let's say that we have some prior knowledge about the energy landscape of this molecule, and let's say that we know that this particular molecule unfolds via two pathways. One pathway is slow, it's slow because it has a higher barrier, and the other pathway is fast, it has a smaller barrier. So now with this extra piece of information, can we tell which of these trajectories came from which pathway? Naively, very naively, one can try to assign trajectories to pathways based on the dwell time. Let's say one can say that maybe the trajectories with the shorter dwell times that unfold very quickly belong to the fast pathway, and those that it takes a while to unfold belong to the slow pathway. What I'm going to do now is to reveal the origin of these trajectories, something we can do in simulations, by coloring them according to the pathway they came from. We can see that the strategy of mapping the trajectories to pathways based on the dwell time is clearly a wrong one. And the reason that the strategy is wrong is that it's very simple. The conformational dynamics is stochastic. And what stochasticity means, among other things, is that there will always be a trajectory, like this red one, that unfolded very quickly, even though it came from the slow pathway. And vice versa, there will always be a trajectory like this blue one that took a while to unfold just because of bad luck, even though these trajectories came from the fast pathway. So what this simple illustration shows is that even when one has access to individual conformational trajectories of biological macromolecules, it generally remains unclear from the trajectories themselves whether the underlying conformational dynamics is mediated by one or multiple pathways. And what this illustration shows is that the issue of identifying multiple pathways is rather fundamental. That is, this is not simply an issue of an insufficient experimental resolution, which makes our central question even more relevant. So how can one tell if the given conformational dynamics are mediated by multiple pathways? We think we found a solution to this problem. Our idea was to try to establish a set, a library, so to speak, of distinguishing signatures in experimentally observable quantities, the signatures that would allow one to tell not only to identify the presence of multiple pathways, but also to tell what particular multi-pathway configuration, kinetic scheme, is mediating the given conformational transition. We approach this problem through a hierarchy of fundamental multi-pathway configurations. The simplest kinetic scheme in this hierarchy, in fact, the simplest imaginable multi-pathway configuration, consists of two pathways, 1 and 2, that directly connect the native state N and the unfolded state U. Without loss of generality, let's assume that pathway 1 dominates the transitions when no force is applied. However, even though pathway 1 is dominant in the absence of the force, the situation may change in the presence of the force, because remember what the force does to the pathways, it changes the barrier height. And so if the barrier of pathway 2, even though this barrier is initially high, if it is softer and therefore more compliant under force, the system will switch from pathway 1 to pathway 2 at a certain force when the barrier along pathway 2 is lowered sufficiently. Now, is there any way to identify this pathway switch in an experiment? We think there is a way. In particular, in the constant force experiment, please focus on the upper subplot, constant force experiment typically measures the unfolding rate as a function of force. In the constant force experiment, a pathway switch in this simple kinetic scheme will manifest itself as an anomalous upturn in the force-dependent rate. Where does this upturn come from? Well, this is simply a consequence of the crossover from the weakly force-dependent rate of unfolding through the stiff barrier to the strongly force-dependent rate of unfolding through the soft barrier. In the force ramp experiment, this is the experiment that measures the distribution of transition forces at different values of the force loading rate. The pathway switch will manifest itself as an anomalous trend in the height of the force distribution, the non-monotonic behavior of the height of the force distribution with increasing the loading rate. The reason I call it anomalous is because this behavior is in stark contrast with what is known for single pathway transitions, where the height of the force distributions monotonically decreases with increasing the loading rate. So to summarize, the distinguishing signatures of a pathway switch in the simple kinetic scheme is an upturn in the force-dependent rate and measurable in the constant force experiment, and the anomalous trend in the height of the force distribution measured in the force ramp experiment. Now, a pathway switch results in an intriguing behavior when one of the pathways resists force. The microscopic reason why this pathway resists force is that the transition state of this pathway is more compact. It has a small extension than the native state. When the force-resistant pathway dominates a transition that's in the absence of force or at low forces, but the force-compliant pathway takes over beyond certain force, then the pathway switch will manifest itself in an anomalous dip in the force-dependent rate. Where does this dip come from? This is simply the result of a crossover from the decreasing rate of the force-resistant pathway to the increasing rate of the force-compliant pathway. Our theory predicts that in the force ramp experiment, the pathway switch from the force-resistant to the force-compliant pathway should manifest itself as the distinct form of bimodality of the force distribution. What's distinct about it is that the low force peak always remains at zero force at all values of the loading rate. Well, this is simply because the low force peak corresponds to transitions through the force-resistant pathways, and the probability of transition through force-resistant pathways is maximum when no force is applied, because it's a force-resistant pathway. To summarize, the distinguishing signatures of a pathway switch from a force-resistant pathway to force-compliant pathway is a dip in the force dependence of the rate, measurable in the constant force experiment, and the distinct form of the bimodality of the force distribution with a zero force peak, measurable in the force ramp experiment. Now let's move up in complexity of multi-pathway configurations, and let's consider an intermediate state sitting on one of the pathways. We considered 1, 2, 3, 4 distinct kinetic schemes with an intermediate. These 4 kinetic schemes differ by the relative rates of transitions of the individual steps in these schemes. In particular, the first 2 schemes correspond to very fast exchange between the native and intermediate state on pathway 2, and the second pair corresponds to the slow exchange between the native and intermediate state. So the first one of these 4 corresponds to the case when the intermediate state is separated from the unfolded state by a large kinetic barrier. As a result, the intermediate state acts as a transient trap. It traps population until this trap is opened up by the force. The consequence of this trapping effect of the intermediate is that the force-dependent transition rate will acquire a plateau. Let's see, I learned this word yesterday, la mesa, right? The plateau, this plateau sets in when the trapping effect of the intermediate becomes evident, and when the trap opens up by the force, the barrier becomes sufficiently small, the plateau ends. In the force ramp experiment, the trapping effect of the intermediate will result in a bimodality of the force distribution that emerges at intermediate loading rate. So it's a transient bimodality. The low force peak in this bimodal force distribution corresponds to transitions via initially fast pathway, and the second peak of this bimodal force distribution corresponds to transitions through the slow pathway with the intermediate state acting as a new source. Alternatively, when the intermediate state is separated from the unfolded state by a small kinetic barrier, the role of the intermediate state is it serves as a gate to the fast transition step. So it's sort of the effect is opposite to the effect of the trap. Our theory predicts that the intermediate that serves as a gate to the fast transition step will manifest itself as a, as a sigmoidal shape of the force-dependent transition rate, measurable in the constant force experiment, and as, again, a non-monotonic trend in the force distribution with the force loading rate, something that one can measure in the forced ramp experiment. Now, so again, these two schemes correspond to fast exchange between the native and intermediate state. In the opposite case, when the exchange between native and intermediate state is slow, the transitions from the native to the intermediate state can be treated as irreversible. The effect of the intermediate state in this case is to sequester the portion of populations, meaning that if you enter the intermediate state, there's no way back. This sequestering effect of the intermediate step manifests itself in yet another form of bimodality of the force distribution, where the low force peak and the high force peak dominate in different ranges of the force loading rate. And so based on this dominance of the two peaks, one can distinguish these two schemes from each other. And finally, an interesting case of multi-pathway configuration is the case where the pathways originate from disconnected native states. When the pathways originate from the common native state, then obviously the net transition rate is simply the sum of the rates of the individual rates along the individual pathways. However, in the case of disconnected native states, the net transition rate in the unfolded state is limited by the slowest pathway. So the slowest pathway becomes the rate limiting step. However, remember, because the force changes the barrier heights, what can happen is that the initially fast pathway can become the rate limiting pathway at a sufficiently large force. Our theory predicts that the multi-pathway configurations with disconnected native states will result in an anomalous downturn in the force-dependent rate. In the force ramp experiment, the multiple native states will manifest itself as yet another form of bimodality, which is interrupted by transient unimodality at intermediate loading rates. Now, where all these scenarios come from? They all come from a unifying theoretical, analytical theoretical framework that we have developed. The foundation of our theory is a Kramer's picture of diffusive escape over the barrier. Our theory results in analytical expressions for the force-dependent rate and for the distribution of forces as a function of loading rate. So in all these plots, the symbols and histograms come from simulations, and all the lines correspond to our analytical theory. And so we can see that the theory is doing a pretty good job reproducing the force-dependent rates and the distribution, the evolution of the force distributions with the loading rate in all these different kinetic schemes, as well as all the distinguishing signatures of these kinetic schemes. I would like to stress that these signatures are truly distinguishing. That is, not only they allow one to distinguish these multi-pathway configurations from each other, but they also allow one to distinguish them from what is known about single pathway transitions. In particular, for a single pathway connecting the native and unfolded state, which has a single activation energy barrier, the bimodal, the force distribution measurable in the force ramp experiment is known to be the height of the distribution is known to decrease monotonically. While in the case of a single pathway, which contains multiple sequential kinetic barriers, the distribution may be bimodal, but the bimodality only emerges at intermediate values of the force loading rate. So everything so far was about the distinguishing signatures that can allow one to identify multiple pathways that govern conformational dynamics. How might biological systems exploit multiple pathways? What our results suggest is that the addition of just one alternative pathway shapes the response of molecules to force in remarkably diverse ways. And this suggests that multiple pathways, through the way they shape the force response, may have important consequences for biological function. For example, in the simplest case of a pathway switch in this simplest imaginable kinetic scheme, what we can see is that the presence of alternative pathway results in the force-dependent rate, which becomes maximized over a broad range of forces. The range of forces broader than what would be possible with either of the two pathways alone. Evidence of this scenario has been observed experimentally in pooling experiments on protein domain SH3, where the unfolding rate as a function of force showed a similar type of upturn at different sets of experimental conditions, suggesting that conformational dynamics, the unfolding of SH3, is likely to occur through this particular multi-pathway kinetic scheme. As a result of the switch from a force-resistant pathway to force-compliant pathway, we can see that the system can readily dissociate at high forces while increasingly resist low forces. This behavior can be readily recognized as a catch bond behavior, which was observed in a number of systems, in particular in leukocytes. This plot is for the lifetime versus force. The lifetime is the inverse of the rate. And so the dip in the rate corresponds to the maximum in the lifetime. And we can see that the signature, the maximum in the lifetime, creates a range of forces at which leukocytes can adhere to blood vessel walls for long enough to roll and search for sites of inflammation. When the intermediate status acts as a transient trap, when it's separated from the unfolded state by a large kinetic barrier, we saw that the system acquires a plateau in the force-dependent rate, which means that there is a range of forces in which the system becomes essentially insensitive to external perturbation. So we can think of it as the emergent robustness at the single molecule level. In the case when the intermediate step acts as a gate to the fast transition step, the force-dependent rate becomes sigmoidal. It's sort of reminiscent of the cooperative binding curves. And indeed, what it allows the system to do is to acquire distinct operating modes. The binding mode with a low off rate and with the dissociation mode with the high off rate. The presence of multiple native states we can see results in a downturn in the force-dependent rate. In other words, it minimizes the force-dependent rate in a broad range of forces. Well, if this is the rate of leaving the functional state, it means that the system can now stay in the functional state in a broad range of external perturbations. Even though not in a pooling experiment, single molecule FRET experiments identified multiple native states in a ribozyme, which may explain why ribozymes can function in a broad range of timescales. So what we can see is that the addition of just one alternative pathway shapes the response of biomolecules to force in remarkably diverse ways. And the resultant scenario may have some important consequences for biological function. So far, I managed to avoid showing any equations, but I hope that you won't mind if I show a few of them, because I think they actually have some practical value. As I already mentioned, our analytical theory is based on the picture of diffusive barrier crossing, Kramer's problem, on a force-dependent or time-dependent potential. Our theory results in analytical expressions for the observable signatures of multiple pathways that we've just discussed. In particular, we were able to derive closed-form expressions for the force at which the system switches pathways. And depending on the range of parameters, this switch will result in an upturn, a dip, or a plateau. And what I think the value of these expressions is that they relate an experimentally observable quantity, the force at which the system exhibits the upturn, dip, or plateau in the rate by switching the pathways, to the microscopic parameters of these pathways, namely the barrier heights, the barrier locations, and the intrinsic rates of these pathways. And as such, these expressions, and our theory has resulted in more of them, provides a direct and practical means of characterizing quantitatively multiple pathways, extracting the barrier and rate parameters of the pathways from the experimental data. Let me summarize the key points here. So the complexity of the energy landscapes of biomolecules suggests that multiple pathways as opposed to a unique single route should be a very common scenario in protein and nucleic acid folding and binding. However, the difficulty is that even when one has access to individual trajectories of biological macromolecules, it generally remains unclear from the trajectories themselves whether these transitions occur through one or multiple pathways. We established a set of model-free, experimentally observable signatures that unambiguously identify multiple pathways. This is true even when the pathways themselves cannot be directly resolved in the experiment. Our theory results in analytical expressions for these observable signatures, and these expressions relate the observable signatures to the microscopic parameters of the pathways. And so one can imagine fitting, I'm talking about simple least square fit of these expressions to experimental data, which directly yields the microscopic parameters of the pathways. And finally, we saw that the presence of even one alternative pathway shaped the response of macromolecules to force in remarkably diverse ways, which provides the systems with a lot of design freedom to tune biological function in response to external perturbation. This work was done by my graduate student who has just graduated a couple of weeks ago, Chris Pierce. I would like to thank our sponsors, National Foundation, National Institutes of Health. I would also like to take this opportunity to advertise multiple research positions in my group for graduate students and postdocs who enjoy scientific challenges. We don't have a shortage of scientific challenges in my group. Our general approach is we are interested in a broad range of biological questions, and I hope to take a few minutes to give an overview of these questions with the approaches from theoretical physics, and our focus is on analytically tractable theory as opposed to purely numerical. So a few questions that we are thinking about hard in my group are the following. So the first question is something that I told you a little bit about today. The idea is that single molecule force spectroscopy is a very powerful tool that enables one to measure so-called mechanical fingerprints of biological macromolecules, or in other words, force extension curves of individual biomolecules. And it is clear that these mechanical fingerprints contain a lot of precious information about the underlying kinetic and energetic parameters of the molecule. And so what we are interested in is in developing analytical theories that would allow one to extract this microscopic information about the landscape, energy landscape of the molecule, about the activation energy barriers, and about the intrinsic rates on these landscapes from the mechanical fingerprints. The second direction in my group is concerned with the structure and dynamics, more precisely structure dynamics relationship in the genome. So this work is done in collaboration with my biology colleague, distinguished immunologist at UCSD, Professor Kees Murray. And what Kees and his group can do is the following. They work with B cells from mice. B cells are the cells that generate antibodies. And in these B cells, there is a region of the genome which is called the immunoglobulin heavy chain locus. I apologize for this terminology. And what this locus does, well, it's responsible for synthesizing antibodies. And in this locus, it consists of gene segments which are called V, D, and J, variable diversity and joining, that doesn't matter. And during a certain stage of the development of B cells, these gene segments, V, D, and J, recombine to form one single gene, VDJ. And because there are multiple V regions, multiple D regions, and multiple J regions, it's a combinatorial process. So one out of, let's say, hundreds of V can find one of the hundreds of J and one out of hundreds of D, right, to form VDJ, right? And because of the combinatorial nature of this process, there are many possibilities in which the segments can form a gene. And this is the key underlying reason why we can generate the number, virtually unlimited number of antibodies. Certainly many more types of antibodies that there are genes in our bodies. So what Kees and his group can do is as follows. They can insert fluorescent markers next to V regions and DJ regions. They can do it in single color experiment, on 2-color experiments. This is a very challenging experiment. They were the first on the planet to be able to do this because the standard biochemical techniques of inserting these markers made these cells unhappy. They just stopped dividing. And so they found an approach where the cells continue functioning normally even with these markers. And so once you have the markers at different sites of the genome, you can then trace them under the microscope, confocal microscope, and record their trajectories. And this is my cartoon of this experiment. So imagine this is a piece of DNA. These two segments, two cylinders mark the locations of V region and DJ region. I'm talking about DJ because when VDJ recombination occurs, it occurs in a certain ordered fashion. First, D and J form DJ, and then V joins DJ to form VDJ. And so these two segments, V and DJ, may be separated by huge genomic distances. I'm talking about mega base pair distances. And yet, in a very timely fashion, then they must find each other. Why do they have to find each other? When in the context of immunology, they have to find each other so that they can recombine. But then there are many processes in biology where this step when distinct gene segments must find each other in a crowded environment of the nucleus is a key step in many processes. For example, these two segments may represent an enhancer and a promoter that must contact each other to activate gene expression. Similar things happen in embryonic development and other critical biological processes. And so what's non-trivial about this is that how do these segments find each other? The task is non-trivial for at least two reasons. First of all, as I already mentioned, these segments may be separated by millions of base pairs genomically. And second, they are moving not through a vessel of water, but they are moving through a very crowded environment. We know that the cellular nucleus is a very crowded environment. And so based on the three-dimensional trajectories, X, Y, Z as a function of time recorded by our biology friends, we try to understand how do the segments move and how do they find each other on a short time scale. What we found, in a nutshell, is that the motion of the genomic segments in the nucleus corresponds to anomalous diffusion, namely subdiffusion, and it is governed by fractional Langevin equation. So for those of you, well, I'm sure all of you are familiar with Langevin equation, right, that describes normal diffusion. The difference between Langevin and fractional Langevin equation is that in fractional Langevin, the viscous term has a memory kernel, and the random force term is no longer that random. There are certain correlations there. And so once we have established the mechanism, fractional Langevin motion, we were able to predict, for example, mean first passage times. So how long does it take for two segments that move according to fractional Langevin motion to find each other? We found that the time scale is impressively short. It can be as short as just a few seconds. And intuitively, this can be understood as in terms of the properties of a subdiffuser. So unlike a normally diffusing particle, a subdiffusing particle does not diffuse as far, right? Because the mean square displacement scales with the time as time to the power of 1/2 and not 1. However, an advantage of being a subdiffusing particle is that you can search locally much more efficiently. So if you think about microscopic trajectories of a subdiffusive particle, it is much more rugged than the normally diffusing trajectory. And so this efficient search means that if the two segments, even though they are genomically far apart, if they are reasonably close spatially, then they have a very good chance to find each other in a very short time. Another factor that determines the mean first passage time, or just the time scale for genomic interactions that we found, is the size of the confinement. So naively, one can say that, well, these segments diffuse inside of a nucleus, so the nucleus may constitute a natural confinement. However, Recently, my colleague at UCSD, Bim Gran, discovered TADS, topological association domain, that we already heard from Sasha about. And these domains can be thought of as the bundles of loops. And the characteristic size of such a domain, well, the size varies, but on average we can think of about a micron. And so this TAD also serves as a natural boundary, natural confinement for the genomic segments. And so from a simple dimensional analysis, one can show that the mean first passage time for a subdiffusive particle has a very strong dependence on the radius of the confinement, namely at the radius to the power of 4, unlike the normally diffusing particle, for which the time scales as r squared. If you're worried about the dimensions, please remember this is anomalous diffusion, and so the units of the diffusion coefficient are micron squared per second to the 1/2. So the dimensions are fine. So these were some of our preliminary findings about the motion of the genome inside the nucleus, and this is a very active direction in my group. And another set of problems that we've looked at is a fascinating phenomenon of biological membrane fusion. We looked at with my graduate student, Yaojun Jiang, who is now at Princeton, we looked at a particular context where membrane fusion occurred, namely viral infection. So as you know, some viruses, such as HIV or influenza, they are covered by lipid membrane envelope. Well, and what this means is that if such a virus wants to infect the cell, it has to somehow fuse with the cell, because cell has its own membrane, right? Now, from the point of view of pure soft matter physics, we know that if we take two membranes, two lipid bilayers, and place them next to each other, it will take forever, meaning days or maybe more, for them to fuse. Well, this is because there is a huge kinetic barrier separating the two layers. I think people are arguing whether this barrier is 60 kT or 100 kT, but both are hopeless because the time depends exponentially on the barrier height. And yet we know that viruses can infect cells on the timescale of a minute. So how is it possible? Of course, biologists already figured out how it's possible on the qualitative level. It turned out that viruses, such as influenza, have fusion proteins sticking out of the membrane, and when these fusion proteins sense a certain trigger, in the case of influenza, the trigger is low pH. In the case of vesicle fusion, the trigger is high calcium concentration. It doesn't matter. Physics is the same. And so once the fusion proteins sense a trigger, they unfold, insert themselves in the target membrane, and pull the two membranes together. So this is a qualitative picture which we managed to frame in again, analytical theory that was published recently. So I think with this, I will stop. Thank you very much for listening.